Welcome to Chapter 8. This section is going to include chromosomes and cell division. Before we jump into how cells make new cells, we have to talk about what happens when cells divide too many times, because it's important to understand that our cells are not immortal. And if they are immortal, it can be kind of a big issue. So the structure that is included on the end caps of all of our chromosomes is known as a telomere. These telomeres are sections of non-coding repetitive DNA that act as a protective end cap at the tip of each chromosome. Now, as the cell divides over and over and over, these protective end caps are going to get shorter and shorter and shorter until they're so short that the cell will go through cell death because it cannot keep dividing. The telomeres are too short. I like to think of telomeres as like a natural expiration date. This is going to be important as you get older because when you are younger, your telomeres are naturally really long because your cells are going to divide so many times. As you get older, your telomeres naturally shorten. So they're not as long when you're older because you don't have to go through cell division quite as much as when you're younger. There can be a few problems when it comes to telomeres. The first is a mutation that causes the telomeres to be naturally shorter at a younger age. So children can be born with Hutchinson-Gilford progeria, which is going to cause the appearance of a much older person, but it's in something like a child or a toddler. And unfortunately, this also means that the life expectancy is a little bit shorter as well, but it is because the telomeres are naturally already starting off super short, as if someone was in their 70s or 80s, but they've just been born. Another problem with telomeres is that some cells are able to rebuild their telomeres. When it comes to rebuilding telomeres, and essentially what will lead to uncontrolled cell division, this is what we call cancer in our bodies. And we'll talk a little bit more about cancer a little bit later in this chapter. So we did touch on this a little bit in earlier chapters in the earlier unit, and that is the difference between chromosomes, or our DNA, when it comes to the two different cell types. Remember that prokaryotic cells are going to be things like bacteria and archaea. Prokaryotic cells are going to have just a single circular chromosome that is going to be floating around in the cytoplasm or the cell gel within the cell. Again, they only have one chromosome and it is circular like a rubber band, if you want to think of it that way. In eukaryotic cells, we are going to have that include animals, plants, fungi, protists. So a little bit more of a variety of organisms that are made up of eukaryotic cells. Within a eukaryotic cell, one of the defining features is that they have a nucleus. The nucleus is going to house the chromosomes, which are going to be linear in their shape. So they are essentially lines of DNA, and there's typically more than one. So typically we're going to see them in pairs of some kind. For example, humans have 46 chromosomes, 23 pairs. The way that prokaryotic cells, so something like bacteria, is going to divide is going to be through a process called binary fission. This is going to cause the exact copy of the parent cell in both of the what we're going to call daughter cells. Daughter cells are going to be the product of any cell division. So what we start with is that parent prokaryotic cell. It is going to replicate the DNA within the cell, so that circular single chromosome is going to make a copy. So technically, at this point, there are two strands of DNA inside of the cell. The cell itself is going to elongate a little bit, and then the two pieces of DNA are going to go to opposite sides of the cell, and then there's going to be a pinching in between the two pieces of DNA, kind of down the middle of the cell, until the cell pinches apart, and you end up with two identical daughter cells. So they are identical to each other. They're also identical to the parent cell that made them. Now, when we get into the process of how eukaryotic cells divide, that's cell division. This is also technically cell division, except this is also how bacteria reproduce. It is a form of asexual reproduction that is going to lead to the addition of individuals within the population of cells. And this is a little bit different from how like we reproduce, right? So this is asexual reproduction. It's essentially cloning. And that's going to be one of the last times we talk about bacteria. When it comes to how eukaryotic cells divide, it really kind of depends on what kind of cell we want to make. Let's start with somatic cells. These are known as body cells, or the cells that make up pretty much any part of your body except for one cell type. We'll talk about it. So this can be like your hair, your skin, your tongue, your heart, your kidneys, your liver, your spleen, your intestines, your blood, so on and so forth. It's all the cells that make up the different tissues in your body. These cells are going to go through mitosis. Mitosis serves the purpose of growth, repair, and replacement of old or damaged cells. In the end, you are going to produce two identical daughter cells. They're genetically identical to each other, and they're identical to the parent cell, and we only get two. Again, this is going to happen throughout your entire body. When it comes to the only type of cell that does not go through mitosis, that is a gamete, also known as sex cells. These are going to be cells that serve the purpose of reproduction. So this is going to come from the process meiosis. Meiosis is going to be a very different process to mitosis because the purpose of meiosis is to produce unique daughter cells. The other thing that's important is the number of chromosomes need to be cut in half because you don't want a sperm and egg cell to have 46 chromosomes. We need a sperm and an egg cell, respectively, to have only 23 chromosomes so that when they fuse, it's going to lead to an offspring that has the total number of chromosomes, half from mom, half from dad. Now, all of these cells, whether or not it is a somatic cell or a gamete, the parent cell is going to have to go through a cell cycle. So this is going to be made up of two primary parts. We have interphase and the mitotic phase. Interphase is where a cell spends the majority of its time. This is going to be growth and normal cell day-to-day life. So performing those normal functions that cell might perform. In the mitotic phase, this is going to be active cell division. So it's going to be the production of a new cell. So in interphase, we're going to have a few kind of parts that make up interphase. The first is gap 1. This is going to be a primary growth phase for the cell. Also, the cell's just going to still keep doing its normal cell functions. In gap 1, this is a big growth period. In fact, it takes up almost half of interphase. From gap 1, we're going to have S phase, or DNA synthesis. This is a very important step when it comes to interphase. This is going to be the part in the cell's life that it is going to replicate all of the chromosomes inside of the nucleus. So S phase, or DNA synthesis, and synthesis means to make something, we are making a copy of all of the DNA inside of the nucleus of the cell. From there, the cell is going to go through another growth phase. This is gap 2. So this is a much shorter growth period as the cell prepares to go into the mitotic phase. Once in the mitotic phase, the cell is going to actively divide into 2 new cells, and then it's going to go through cytokinesis. Now, technically speaking, cytokinesis is at the very, very end of mitosis, but it is not part of mitosis. It is going to be part of interphase. Cytokinesis is going to be the movement of the cytoplasm, the cell gel, between the 2 cells. So it's kind of like dividing assets between the 2 daughter cells. Now, between all of those important parts of the cell cycle, there's going to be a control system. This is going to be kind of a checks and balances to make sure that the cell is performing those functions it needs to complete before going into the next step of the cell cycle. So the first is the G1S checkpoint. This is going to check and make sure that the DNA is not damaged. If it is damaged, then the cell might go through cell death, because we don't want to go into the next checkpoint or past the next checkpoint, because that would go into mitosis, and we don't want damaged DNA to be replicated and put into other cells. The next checkpoint is going to make sure that the DNA is replicated properly. If it is not, the cell might go through cell death, or it might just try to go back in order to complete DNA synthesis. Once the DNA has properly replicated, we then go into mitosis. That last checkpoint is less important to me, but it's just kind of making sure that certain things have been completed in that mitosis. From here, we're going to look at that specific part of interphase, which is DNA replication or DNA synthesis. So again, this is going to happen during the S part of interphase. Very important, it happens before mitosis. We're going to have a parent DNA molecule. This is going to be like the DNA original strand. We are going to unzip the parent DNA so that double-stranded DNA, we have one on either side. We're going to fill in the complementary side of the DNA strands so that a little bit of the original DNA molecule is in the 2 new DNA molecules. So this is a nice overview of how DNA replication happens. So the first step is unwinding and separation. This is going to happen from an enzyme called DNA helicase. DNA helicase is going to unwind the DNA. It is separating the 2 original DNA strands. From there, another enzyme is going to come in the second step, where we have reconstruction and elongation. It is called DNA polymerase. DNA polymerase is going to be like a builder. It is going to build that other side of the DNA. So we have the original DNA strand being unwound, and then DNA polymerase comes in and builds the other side of the strand on both sets of the original DNA. So we get an exact copy of the original DNA strand. So during this process of DNA replication, there can be problems or errors. If you were to try to rewrite all of the notes that you've taken right now and just freehand write it, you'd probably make some mistakes. That can also happen during this process of DNA replication. Mutations are what we call these errors. Anything that is a change to the nucleotide bases of the DNA, meaning our A's, T's, C's, and G's, is called a mutation. So why would errors during this DNA replication potentially be a good thing? And the answer, guys, is going to be evolution. Mutations are quite literally the basis for evolution, and we'll get to that chapter eventually. But for now, just know not all mutations are bad. They can be helpful, they can also just be silent. So not all mutations are detrimental. Okay, so let's start with mitosis. Remember that mitosis serves the purpose of creating genetically identical cells, and it's required for growth replacement and repair of potentially damaged or old cells. So when a cell reaches the end of its life, either it's too damaged to go on, it's replicated too many times, it goes through a process called apoptosis. This is also called programmed death of a cell or programmed cell death, however you want to think of it. But essentially, it's like the cell is retiring. It is no longer going to do the job or function it originally had, and it can't keep producing new cells. So you have a handful of different organs and tissues that do have to experience apoptosis a little bit quicker than others. We'll talk about that rate of replacement here in just a second. Now, I do like to show you guys a picture of a skin slide. So this is a section of skin, and where you see that topmost layer of that dark purple, that is the upper layers of your skin. Now, you'll notice that the very top layer is going to have kind of like stringy bits coming off, and that is skin that is sloughing off. You are constantly shedding skin all the time. This is important because it is dried out, keratinized skin, and fresh, new, hydrated skin is underneath that drier skin. Now, the reason that this happens is that bottommost layer of the dark purple cells, it's going to go through mitosis where we are making new cells, and those new cells are pushing up on that bottom layer and pushing the cells on the top off. I kind of like to think of it like that ice cream treat, like a push pop. You are pushing from the bottom and the top is coming off as you consume the push pop. Same kind of concept here for your skin. So when you think about what dust is, it's like 98% skin cells. So kind of gross, makes you want to dust your house. I said we'd talk about rates of mitosis, and here it is. When cells go through mitosis, it can happen at a different pace. So for example, your red blood cells replace themselves every 2 to 4 months. This is kind of a slower rate of replacement, not the slowest tissue in your body, but also not the quickest. One of the quicker cell types that is going to replace itself very quickly is the lining of your intestines. This is going to happen roughly every 3 weeks, and that is because those cells are constantly being exposed to acid as well as maybe abrasions, so they're getting damaged pretty quickly. We don't want those cells to stick around and end up causing a problem. So those cells will go through apoptosis and be replaced by new cells every 3 weeks. Before mitosis can begin, remember that the DNA, our chromosomes, have to replicate. So if you look at the right, we are looking at a single chromosome at the top, that line of DNA. It is going through replication. Once it goes through replication, we are going to call the chromosome a sister chromatid. And that sister chromatid is going to be attached together at a central hip point, which is called the centromere. Now, as mitosis begins, the sister chromatids are going to start to condense. It's kind of like the DNA is tightening up, flexing, if you will. And the reason being is that we need the sister chromatids to condense so that when we eventually rip them apart, they're going to rip apart evenly. As mitosis begins, the first part of mitosis, we're going to see spindle fibers form. And the spindle fibers are going to do a bunch of work for us here in pulling those sister chromatids apart. All right, now we are into the four-stage process of mitosis. Now, before we can get into the actual steps, we do need to talk about that interphase real quick. Remember that in interphase, that is when DNA synthesis is going to happen. DNA synthesis is not part of mitosis, and neither is interphase. It has to happen before mitosis can begin. So our chromosomes have turned into sister chromatids. There's an extra copy of DNA that is still attached to each other at the centromere. This is very important. So what this means is that our sister chromatids, as long as they're connected, they are considered one chromosome. From here, we jump into the four stages of mitosis. You can remember them with the acronym PMAT, P-M-A-T. We are starting with P, that is prophase. In prophase, there are four things that are going to happen. The first is that the nuclear membrane starts to break down. So it is the membrane that surrounds the DNA. The sister chromatids are going to finish condensing. A centriole, which is a type of organelle in the cell, is going to produce spindle fibers. And the fourth thing that happens, I do want you to know, is that the centrioles, which are producing the spindle fibers, are moving to opposite ends of the cell. This leads us into the second stage, which is metaphase. The centrioles have now moved to opposite ends of the cell, and the spindle fibers have attached to the centromere, that centermost point, of the sister chromatids. Now, the centrioles, with their spindle fibers, are going to play a game of tug-of-war. They're going to keep pulling back and forth, back and forth, back and forth, until the sister chromatids line up across the middle of the cell. So when you hear metaphase, think middle, middle. All right, after metaphase, we have anaphase. Anaphase is going to be where that game of tug-of-war pulls the sister chromatids apart, where one full set of chromosomes is being pulled to one side of the cell, and then the other set is going to be pulled to the opposite side of the cell. Now keep in mind that the sister chromatids were identical copies of DNA. So there's an identical copy going to one side, an identical copy going to the other. After anaphase, we have telophase. In telophase, you're going to see the chromosomes, because they're no longer sister chromatids, the chromosomes will start to relax or uncoil. A nuclear membrane will start to reform around both sets of DNA, and the spindle fibers are going to start wrapping around the middlemost point of the cell and cause a pinching in of the cell. I like to think of those videos of a watermelon getting wrapped in a rubber band over and over and over until eventually the watermelon pops. And essentially that's what happens at the end of telophase. Those spindle fibers wrap around and around and around until the cell pinches apart. And that is the last stage of mitosis. Because remember, cytokinesis is just going to be the movement of the cytoplasm into each of the new cells. So it's again like dividing the assets between those two cells. The process we just went through is how our cells make new cells. However, there can be a situation where cells go through mitosis too much. This is going to be a mutation where the cell divides out of control. That is called cancer. This is a very serious topic, a very serious health issue. It's the second leading cause of death in the United States, the first being heart disease. So cancer is responsible for 20% of all deaths worldwide. This is a big issue. There are multiple types of cancers. They work in different ways and they can cause different problems. But we are going to talk about the three primary concerns when it comes to cancerous cells. Cancer is different from our normal cells in three ways, and they are the three ways that cancer is a problem. The first is that cancer cells have no contact inhibition. This means that cancerous cells are going to fill a space and keep growing and growing. It's kind of like being impolite or rude because they're pushing, as far as those cells go, on our tissues. And this becomes a really big issue and one of the reasons why cancer can kill you. It is because the cells will basically cause that pressure on our tissues, organs, and cells, and it leads to our cells not being able to perform the functions that they normally would. Normal cells would fill a space and stop growing. They would not push on other tissues the way cancer cells can. Cancer cells also divide indefinitely. So where our cells have a natural expiration when their telomeres get too short, cancer cells can regrow their telomeres and divide nonstop. They don't have to quit. They're essentially immortal cells. The last is that cancer cells have a reduced stickiness. Our cells do not flake off, and you start growing a heart cell and then a new heart somewhere. Cancer cells can shed and spread. This is a pretty big issue when it comes to cancer. So cancer cells can break off from that primary clump of cells and start growing in a new place of your body. Okay, we've probably heard the term tumor before. Cancerous tumors can be a pretty big issue because they can do all of the three things that we just mentioned that leads to problems with cancerous cells. There are two types of tumors that can still be cancerous, and that is benign tumors, which can refer to tumors that can be removed safely, potentially that they're not growing, or in an area where they're not causing a whole lot of problems. That doesn't mean it's a good tumor. That just means that it is not causing issues like a malignant tumor might be. Malignant tumors are going to be inoperable. They can also be in an area that is causing a lot of problems. You can think of like maybe your brain stem or your brain or your spine. Malignant tumors can also shed and spread. This is going to be called metastasizing. Now, the reason that cancer and tumors can kill an organism is that the tumor is going to cause the tissues, the healthy tissues of our tissues surrounding the tumor, from being able to perform their normal functions. It might cause the cells to die, but essentially it starves our cells for the nutrients that it needs. The tumor ends up consuming those nutrients, and essentially the cancer doesn't kill you. It is the cancer preventing your cells from doing their normal functions that kills you. There are two primary treatments for cancer, and they're both pretty aggressive. The first is chemotherapy. This is going to be drugs that are going to interfere with mitosis. So it is going to attack one of the primary areas that makes cancer so dangerous, its ability to reproduce and reproduce quickly. Now, because of that, this chemotherapy is also going to affect your healthy cells. This is why patients who get chemotherapy usually lose their hair. It's because the cells that cause your hair to grow, that go through mitosis, they go through mitosis pretty quickly. So this chemotherapy is not going to discriminate between the cancerous cells and your healthy cells. And that is why people lose their hair. They also have digestive problems because remember, your intestines are going to go through mitosis pretty quickly because we have that high turnover rate. So chemo also affects the digestive system pretty aggressively as well. That's why people who get chemo have a tendency to lose weight. The other treatment is radiation, and usually you have chemo and radiation together to make sure that you get all the cancer. Radiation is going to be more targeted, where chemo is going to be widespread throughout the body. Radiation is going to be maybe a pill or something kind of like a laser that is going to target and basically irradiate the cancerous cells in a particular spot. So we might use radiation on a tumor in tandem with chemo. We're trying to reduce the size of the tumor and kill the cancer. Another way that we can treat cancer is just by surgical removal, but not all tumors can be removed surgically, or they're too large to remove, or in an area that we can't remove. And remember that the cause of cancer is a mutation that causes those three problems, that contact inhibition, dividing indefinitely, and reduced stickiness. So it is a mutation in the cell itself that is going to cause those three potential things to happen within that cell. Before we get into meiosis, we do need to talk about a few things first. The first is an overview of sexual reproduction, and kind of this is going to lead to the why meiosis is the way that it is. So for asexual reproduction, like what happens with bacteria in binary fission, that is going to be a single parent producing an identical offspring, or you could write down the clone of itself. So that's going to lead to organisms that are identical. With sexual reproduction, this is going to require two parents. They both need to have a gamete, so typically a sperm and an egg. Those reproductive cells are going to fuse together in a process called fertilization, and that fertilization is going to produce something called a zygote, a Z-Y-G-O-T-E. I apologize, it's not on this slide as of the moment. I might add it in afterwards. So that zygote is the combination genetically of both parents. So in order to have that combination, you need to have half the number of chromosomes in the sperm and the egg, so that when they fuse together, they're a nice little mismatch of those chromosomes coming together to make a fully chromosomed, which is not a word, organism. We'll talk about it. So these are going to relate to some terms we're going to discuss here in just a little bit. And it just so happens that little bit is right now. So meiosis is going to produce cells like a sperm and an egg cell that are half the number of chromosomes from the original parent cell. So for example, the parent cell in this case would be 46 chromosomes. In order for the sperm and egg cell to be half that number, it goes through meiosis. What we call this number when it is half is haploid. So gametes are going to be haploid, half the number of chromosomes that we start with in the parent cell. The parent cell is going to be called diploid. The di part of diploid means two. So there are two sets or two copies of chromosomes in that cell. So that parent cell is going to be diploid. That is going to produce a haploid gamete. And then when our egg and sperm fuse through fertilization, you get a diploid zygote that leads to a diploid offspring. Okay, meiosis is going to have two important outcomes. We do want to reduce the amount of genetic material for the gametes, so for the sperm and egg cells. We need to cut the number of chromosomes in half. The other thing that is going to be very important as an outcome for meiosis is that it produces gametes that are going to differ from one another. However, that being said, from a parent, so like a mom and a dad, there's only so much recombination that you can have. Now, what this is going to lead to is diversity amongst the offspring that those two individuals have together. This is still going to end up appearing as traits that repeat. So whether they repeat within siblings, like the Culkin siblings, or the Culkin brothers, they are going to have features that they share because they have the same mother and father. There's only 46 chromosomes from mom and 46 from dad that you can recombine into 23. So you do have the repetition of various features that end up making it to where siblings have similar features, they share those traits, and you know that they're related. Because sperm and egg are produced in the gonads, that is going to be the only place that meiosis occurs. Only in the gonads, so this is going to be in the testes or the ovaries. Meiosis, again, is going to start with a parent cell that contains 46 chromosomes, and by the end of meiosis, they will have 23. So that the offspring is going to have a maternal and paternal copy of each chromosome. What we call this is a homologue. Now, we have 22 homologous pairs of chromosomes. So you have 2 chromosomes 1, 2 chromosomes 2, 2 chromosomes 3, 2 chromosomes 4, all the way up to 22. And then you have an additional sex chromosomes. This is going to be an X and an X or an X and a Y. Now, when it comes to these homologous pairs, again, you have one copy from your mom and one copy from your dad. Maternal means mom, paternal means dad. Now, I do believe I asked you to draw this out. This is the image I want you to draw. You can pause it here. You should label everything. Draw both the maternal and paternal copy. Make sure to draw the sister chromatids, label the maternal and paternal copy, indicate that it's going through replication, and identify the centromere. You can pause this slide here if you need a second to draw. This is going to be the process of meiosis that is really simplified. So the starting point is going to be a diploid parent cell, meaning we have all the numbers of chromosomes. In the human example, this is going to be 46 chromosomes. So you have 23 maternal, 23 paternal. That's going to be your homologous pairs. So that is that starting point. That starting point is going to be interphase. By the end of interphase and the start of meiosis 1, you're going to have sister chromatids for each chromosome. So we still have to go through that same process that we did with mitosis. Meiosis is going to be the same. We have to go through interphase. That is normal cell growth, as well as DNA synthesis. We got to make a copy of the DNA. So once we start with meiosis 1, this is going to focus on the division of the homologous pairs. Underline it, highlight it, put a star next to it. Meiosis 1, the first part of meiosis, is going to start with a diploid parent cell. We have sister chromatids, DNA synthesis has already started, and what separates is the homologous pairs. So the maternal and paternal copies separate. By the end of meiosis 1, we have 2 cells and one is going to contain one half of the homologous pair. The other contains the other half of the homologous pair, and I'm sorry if that was really loud. I just hit the microphone. I talk with my hands a lot. I apologize. So we have two cells at the start of meiosis II. The only thing present is the sister chromatids. So at this point, guys, we're going to separate the sister chromatids. That's the only thing we can separate. So at the end of meiosis II, we have 4 unique haploid, because it's half the number of chromosomes that we started with, daughter cells. This is how gametes are produced. So sperm and egg cells. We have 4 unique haploid daughter cells. Okay, let's get into the process of meiosis. So of course we start with interphase. Remember, in interphase we have normal cell functions happening and DNA synthesis. So our DNA, our chromosomes, replicate into sister chromatids. That is the same as mitosis. Meiosis is divided into 2 primary parts. This is the first. So this is meiosis I. A very important thing to make a note of and put a star next to is the homologous pairs are separating. The homologs separate. Now, I have kind of squared out all of the parts in red that are different from mitosis. This is specific to meiosis. You will need to be able to match the definition to the term as far as like prophase 1, metaphase 1, anaphase 1, so on and so forth. So you need to know what's happening in each of the stages of the parts of meiosis. So let's start. Prophase 1. There are 6 things happening. 4 of them are the same as mitosis. The chromosomes, the sister chromatids, condense. The spindle fibers form, the nuclear membrane breaks down, and the centrioles are moving to opposite ends of the cell. The difference is that the homologous pairs are going to form a structure called a tetrad. That tetrad is going to have something funky happen called crossing over. This crossing over is going to cause part of the chromosome to swap with the other homolog. We'll talk about this a little bit more here in just a bit. Just know that in prophase 1, the homologous pairs form a tetrad and cross over a little bit of DNA. That leads us into metaphase 1. The big difference here is that it's not the sister chromatids lining up, it's the homologous pairs that are being moved into the middle of the cell. It's the same process. The spindle fibers are doing that tug-of-war situation and pulling the homologous pairs that are still in a tetrad into the middle of the cell. In anaphase 1, the spindle fibers are ripping the homologous pairs apart. At this point, we have an uneven separation between the chromosomes. In telophase 1 and cytokinesis, we end up having kind of the same thing happen as we do in mitosis. The chromosomes will start to relax. The nuclear membrane is going to reform around the chromosomes. You have that pinching in through the middle of the cell until it pinches apart, and then the cytoplasm is going to be divided between the 2 new cells. Now, before meiosis II can begin, there's a very brief interphase that is going to happen where the cell is going to grow just a little bit and the sister chromatids are going to start to condense. That leads us into meiosis II. The important part here to remember is that the sister chromatids are separating. The homologous pairs are already separated. They are not together anymore. And at this point, the cells that we're looking at are going to be haploid. they are no longer diploid. They are already at half the number of chromosomes that we started with. And we're looking at 2 cells. But essentially this process is going to be almost identical to mitosis. So in prophase 2, all the things that happen in mitosis are also happening here in prophase 2. We have the sister chromatids condensing, spindle fibers forming, the centrioles are moving to opposite ends of the cell, and the nuclear membrane is starting to disappear. In metaphase 2, the spindle fibers are going to attach at the centromeres. They're going to play that tug of war and pull back and forth until the sister chromatids line up at that middle point in the cell. From there, we have anaphase 2. This is going to be where that game of tug of war rips the sister chromatids apart. So this is the separation of the sister chromatids. And then last but not least, we have telophase 2 and cytokinesis, and that is going to be the pinching in between those 2 cells in order to pinch them apart, and we get 4. Within those 4 cells, the chromosomes start to relax and uncoil, and a nuclear membrane starts to form around the DNA. Woo, my mind just went blank for 2 seconds. At the end here, we have 4 unique haploid daughter cells. So we start with a diploid parent, and through meiosis 1 and then meiosis 2, we end up with unique daughter cells, and there are 4 of them. Now, up till this point, I've been talking about sperm and egg cells as if the whole process is perfectly equal and the same thing. Unfortunately, it's not. For males, they will be able to produce sperm cells from puberty until the day they die. And through the process of meiosis, 4 sperm cells are produced every time. For females, the process of meiosis is going to result in 1 viable egg cell. And it's because through meiosis 1 and meiosis 2, we're going to end up with something kind of like a trash cell called a polar body. These are just going to be reabsorbed into the body, but they don't result in a viable egg cell. What this leads to actually is that the egg cell is going to be massive. Now you can see kind of a size comparison up in the upper right corner. The little blue guy is a sperm cell. The big pink thing that looks like a planet is the egg cell. And in fact, human egg cells are so massive that you can see it with the naked eye during ovulation. It is a very neat thing that this is such a large cell. In fact, it's one of the only cells, aside from like maybe chicken eggs, that type of thing, that you can see that individual cell with the naked eye. But sperm cells are too small to see with the naked eye. So a little bit of a difference between male and female gametes. Okay, quickly talking about that crossing over process with our homologous pairs. So remember that this happens in prophase 1 of meiosis 1. So the maternal and paternal sister chromatids are going to form a tetrad. This tetrad is going to be like a bear hug, if you want to think of it that way. And in this bear hug, there is going to be genetic recombination occurring. We call this crossing over. It is when homologous pairs swap a little bit of genetic information. Now remember, because it's a maternal and paternal homologous pair, these are both like chromosome 3 or they're both chromosome 7. It doesn't really matter as long as they are a pair, because that genetic information that they're swapping is essentially that same information on both chromosomes. It just might be different alleles. because remember, alleles are just variations of a gene, but we're swapping just a little bit. And what this causes is genetic diversity, or you can think of it as variation within a species. Where this crossing over occurs on the chromosome is the chiasmata. So it's just the exchanging point. So now that we've gone over binary fission and that process of meiosis and what it leads to, I want to kind of give you a thought process of sexual versus asexual reproduction and which one might be better. So bacteria, as we know, we have discussed it in the capacity that every cell is identical to its parent cell and identical to each other. So within a population of bacteria, they're going to be all genetically the same. There's no variability. This is actually one of the shortcomings of something like bacteria, because if we introduce an antibiotic that kills that species into, say, like a petri dish that we're growing of the bacteria, the entire population is going to die because there's no genetic diversity. However, a benefit of reproducing like a bacteria, which is asexual reproduction, is it's quick, easy, and there's no risk from mating. So you don't have to put in the energy to find a mate. You don't have any potential repercussions like being vulnerable to predation because you're mating. It's got a lot of benefits. When it comes to sexual reproduction, however, this is going to be like plants and animals, reproducing can be a little bit not as beneficial. The biggest thing we've got going for us is its genetic diversity, because you are combining the gametes of the two parents into the offspring. We are getting a lot of genetic diversity so that something like a disease, if it wipes out part of the population, it's probably not going to wipe out everybody because we have that diversity amongst us. However, there's a lot of cons, like predation from a mate. So something like spiders, typically the female will eat the male after mating. There's also vulnerability to predators during mating, because during that act, you're a little vulnerable. Disease from mating, so you might get parasites or various diseases from your mate. And then there's a lot of energy expended in finding a mate, as well as the reproductive process itself. So is one method better than the other? In my opinion, no, because we see species that have asexual reproduction. We see other species that have sexual reproduction. So since we still see plenty of species doing both, neither one is better than the other. There's just pros and cons to each. Now, kind of continuing on with reproduction as well as meiosis. Sex determination is going to be a little bit different in humans than it is in a few other animals. For humans, we inherit through that process of meiosis and then fertilization from the gametes. We inherit sex chromosomes from our parents. Your mom will give you an X chromosome because females have an XX combination for their sex chromosomes if they are female. Males will have XY. What's very important here, guys, is the X chromosome. There is no important genetic information really, as far as important for development of a functioning human being on the Y chromosome, but you must have a minimum of one X chromosome. Everybody has one. You have to have it. I kind of showed you a picture there on the last slide. The X chromosome is massive compared to the Y chromosome. It's very important, and it contains more than just information that determines your sex. There's a lot of other genes on the X chromosome that are completely necessary to create a functioning human being. How it's inherited, again, is from your parents. Your mom has 2 X chromosomes. That means that in her egg cells, there will be 1 X chromosome because you have a 50-50 chance of getting 1 X versus the other. For your dad, his sperm cells will either have an X chromosome or a Y chromosome because that's what he has, and that is what he can offer from his genetics, so either an X or a Y. So during fertilization, the sperm cell, either carrying an X or carrying a Y chromosome, are going to fertilize the egg cell, and that leads to the sex of the offspring. So, in cases of individuals who are maybe a little upset that they did not have a boy or a girl, it's always the dad's fault. If you want to think of it that way, the sperm determines the sex of the offspring. So it is the dad's fault. Other organisms might determine their sex in other ways. Birds, a lot of females are able to determine the sex of the offspring. This might be through observations that maybe there's not enough males in the population, and then they will decide to have male offspring to offset the population. Ants, bees, and wasps are going to determine their sex by the number of chromosome sets. So females will have a different number of chromosome sets than males. And also in most of these species, ants, bees, and wasps, most of their population is going to be female. The males are typically only used for reproductive purposes. So whenever you see one of those bugs anywhere, it's probably a female. When it comes to turtles, as well as a handful of other reptiles, the environment is going to determine the sex of the offspring. So whether or not the eggs are warmer or colder, temperature determines whether or not it'll be a female or a male. And we do have things called hermaphrodites. Hermaphrodites are going to be organisms that have both male and female gametes. Now, humans are not considered hermaphrodites if they have both sets of genitalia. This is called intersex, so it's kind of a between sexes. And most individuals who are intersex usually have some type of, during development, a hormone malfunction or a mutation of some kind, and they typically will not produce gametes. But actual hermaphrodites might be something like earthworms, garden snails, most plants, and my favorite is flatworms. Now, we can look at chromosomal disorders potentially prior to birth. This might be important for parents or family planning purposes. But if there is already a fetus that is developing inside of the womb, you can look at their chromosomes as well as potentially the sex of the fetus at an earlier stage if you don't want to do an ultrasound. And that is through one of 2 processes. So either amniocentesis, where we take a sample of the amniotic fluid, that is the fluid that is surrounding the fetus, and it's going to be full of cells from the fetus, but this is going to require a large needle that is going to have to penetrate the placenta and also penetrate maybe through the cervix or various areas depending on how we're going into the placenta. So think a big needle. The second type of testing is chorionic villus sampling, or CVS. This is going to also require a big needle, but it's not going to have to go into the placenta to the fluid that's around the fetus. This can be obtained by taking a sample of the placenta itself, which is going to be composed of cells from mom and also the fetus. Chromosome disorders that you might test for, looking at a karyotype of a fetus, might be something like Down syndrome. This is a condition that was first described by Dr. John Langdon Down in 1866. This is the additional copy of chromosome 21. About 1 in 1,000 children are affected by trisomy 21 or Down syndrome. And there's a handful of other chromosomes that you can have an additional copy of that does lead to viable human life. Those would include chromosomes 13, 15, 18, 21, and 22. Any other chromosome, if you had a third copy of it, will not result in a viable human life. So how this occurs is through non-disjunction. During meiosis, something does not split properly, and you are left with too many chromosomes in the gametes. So this is, if you want to think of it this way, unequal distribution of chromosomes during cell division. And it can happen in meiosis 1 or meiosis 2. It just depends where that non-disjunction or unequal distribution might occur. I do have a few examples that I'd like you to write down as far as chromosomes that you might have too few or too many of and what it causes. So you need to know the syndrome name. You need to know what it affects, either a male or a female. I want you to know the genotype and be able to give a short description. So our first condition is Turner syndrome. This affects females. It is going to be only one X chromosome. This might be an X that's attached to the egg cell and nothing attached to the sperm cell from the parents, or vice versa. There's an X chromosome on the sperm cell, but none on the egg cell. This is going to result in females that have shorter height, webbing of skin between their neck and shoulders. They might have underdeveloped ovaries, and they are often sterile. They might also have learning difficulties. Another condition I want you to know is Klinefelter syndrome. This is going to affect males. This is going to be the addition either on the the egg cell or the sperm cell where there's an extra X chromosome. This leads to individuals that have XXY. So they have an extra X, but they also have a Y. This is going to be males that have underdeveloped testes, lower testosterone levels, which is going to lead to that underdeveloped testes, as well as potentially infertility. Oddly enough, they have a tendency to be pretty tall and have very long limbs. They might also have more feminine features, potentially like breast or more of a curve to their hips and thighs. The next condition is called an XYY male, sometimes called super males. This is going to be an extra Y chromosome on the sperm cell that is going to lead to these individuals. They have a tendency to be taller than average, maybe have severe acne. They might have intelligence issues or lower than average intelligence, and potentially larger teeth. The other thing that can happen is that these individuals have a tendency to have too much testosterone and occasionally have kind of an inability to make informed decisions. So kind of a lower inhibition, if you want to think of it that way. Our next condition is XXX females. This is the addition of an X chromosome. It could be on the egg or the sperm. Either way, these individuals do not have any obvious physical or mental signs, but they might be sterile, so unable to have offspring. Sometimes they're called metafemales. Otherwise, there is no noticeable physical features that seems to be a result of having that extra X chromosome. And with that, guys, it is the end of chapter 8. If you have any questions at all, please let me know. Otherwise, happy studying, and I'll see you for chapter 9.